Hello everyone, today we are discussing the page number 62, embryonic development of first few weeks in this lectures of reading book Anatomy for dental students. Chapter contents: Introduction, fundamental processes underlying development, the first 3 weeks of development, folding of the embryonic plate and its consequences. Introduction. Embryology is a fascinating subject and is the foundation of the development, growth and maturation of all the cells, organs and tissues of the body. Strictly, embryology is the study of the early processes of development, beginning at fertilization and following the processes that turn a single cell into a multicellular organism. It is all about generating or building blocks required to make a human body. Developmental anatomy is the study of how these building blocks are turned into specific cells, tissues, and organs, as well as the general growth of the body, as you will soon appreciate in the following paragraphs. All organs and systems do not develop at the same rate, so there is a degree of overlap between embryology and developmental anatomy. For example, the heart and circulatory system must develop and be functional very early in development to ensure adequate supplies of nutrients to the developing fetal tissues. Teeth, on the other hand, are not going to be used until about 6 months after birth at the earliest. While the heart is already beating away, each developing tooth is merely a tiny group of cells bearing little resemblance to a fully formed tooth. Human gestation is considered to take 9 months. More accurately, it usually lasts for 38 to 39 weeks from fertilization to birth. Human 9 months, 38 to 39 weeks from fertilization to birth. Clinically, it is divided into 3 trimesters of 3 months each. In this chapter, we will focus on events in the first few weeks. During the first 2.5 weeks after fertilization, the very basic building blocks are formed from the single fertilized cell. This is a pre-embryonic period. The embryonic period covers the next 5.5 weeks, during which these basic building blocks develop into the cells. tissues and organs. As already indicated, some of these may be in a very rudimentary state at the end of the embryonic period. The remaining 30 or so weeks is the fatal period when the tissues and organs of the body grow and develop, and the fetus grows considerably. We are not fully mature organisms at birth and have another 20 years growing. This chapter is an account of the development of the embryo from fertilization to about the middle of embryonic period, where all the rudiments and building blocks of significance to dental students are formed. The subsequent development of specific systems, the circulatory, respiratory, nervous, and structures forming the head and neck are dealt with in the corresponding section describing anatomy. The importance of understanding development. Understanding the very early stage of development provides a general knowledge of how you develop and will equip you with a vocabulary to understand the terms and processes that takes place during the development of these systems relevant to dental practice. Fundamental processes underlying development. Embryology is essential. The development of all the different types of cells of the body from a single cell. It follows, therefore, the cell division or mitosis is a major process in development. However, cell division also needs to be limited in certain areas at certain stages of development. Otherwise, each cell type would be equally as numerous. Your teeth would be the same size as your brain. The rate of mitosis in one tissue also needs to be coordinated with the cell division in another tissue so that structures develop in harmony. Cell death is another important process in embryological development. It may seem strange to introduce death into a chapter about being of live. Cell death during development is not random, but is programmed. Cell death or apoptosis, in which cells are programmed to commit suicide at specific times. Knowledge of the way in which different structures develop often clarifies the mature anatomy, especially the overall patterns that govern the way things are arranged in the body. Embryology can give a logical explanation of many anatomical where there appears to be none. The embryology or developmental anatomy of a given structure can often explain apparent anomalies or illogicalities. Why is the diaphragm innervated by nerves that originate in the neck? Why do nerves innervating the larynx go all the way down into the thorax and come all the way back up into the neck, when it would be much simpler to direct the nerves straight across the neck to the larynx. The mysteries of the strange courses of the phrenic nerves and recurrent laryngeal nerves will be explained in context of chapter 13 respectively. As you read the description in this and other chapters, covering development, you will see that embryology and development is an awe-inspiring process. You will wonder how anybody ever arrives in this world with 20 tiny fingers and 20 toes on those little limbs, those eyes and rosy cheeks, where with life being all us, many individuals do not. Something has gone wrong during their embryological development to cause the absence or deformity of tissues or organs. Such developmental anomalies may be life-threatening at worst, even at best. Many will cause functional and cosmetic defects, render life difficult. As practicing dentists, you will see abnormalities on the face, jaws, teeth, which you may be able to correct yourself or refer to another colleagues specializing in their treatment. You'll be expected to understand how such conditions arose, if known, as many parents will be at least curious and may be very concerned about the consequences and possibility of risks inherited by their children. You will also come across records of abnormalities in other systems during the taking of patients' medical histories. Some conditions will affect the way in which you treat your patients and must be taken into account during your treatment planning. The most dangerous period of development to arise is the pre-embryonic period. The embryonic period is only marginally less dangerous and still fraught with hazard. The fetus is not entirely safe until delivered at the end of the gestation. The risks would be borne in mind when treating pregnant patients. The use of many drugs and chemicals is very strongly contraindicated during pregnancy, as they can cause developmental defects. Two examples of the role played by apoptosis are the formation of hollow tubes from solid cord of cells, or the removal of tissues that would restrict the movement and development of other tissue. Movement of cells in another absolutely vital process in the developing takes place at several levels. Cells have to change shape to bring about a specific configuration so that their functions are maximally efficient. Coordinated change of shape in all cells across a sheet of tissue can alter the shape of the tissue from a flat sheet into a curve or even a tube. If cells simply accumulated as a result of multiple cell division, we would end up as a large ball of cells without any recognizable form. Cells have to move as individuals or populations along defined pathways to produce definitive shapes and structures. This is cell migration. Often, one population of cells may be required to migrate while another remains static. Ultimately, millions of cells are going to arise from one single fertilized ovum. It may be surprising that only about 200 different basic cell types, recognizable by their different shapes and functions, make up the human body, although some cell types are further subdivided by functional chemical differences, but cannot be seen directly with microscopes. Tissues are aggregates of cells, usually comprising several different cell types. Cell must therefore use the repertories outlines in the previous paragraphs to divide and migrate to the correct location at the right time. Ultimately, tissues are combined to form specific organs and systems by further division, migration and combination. You will already be aware that most cells in the body have specific function. When cells are fully differentiated to carry out their specific function, most cannot change their function at all. Certainly not radically. Fat cells cannot be used as neurons, and muscle cannot be bone. That creates this difference between these cells. All cells within the body contain the same genetic information, with the exception of red blood cells that lack nuclei and the gametes, ova, and spermatozoa, the exception. So that only contain half the genetic information. So why is a neuron different from a adipocyte? It is all due to gene expression. The specific genes that are expressed within each cell type, that means each one unique from another cell by determining the types of proteins that a particular cell will produce. The proteins in turn determine the shape and function of cells, how they attach to each other and other cell types, what type of receptors they carry, and therefore what messengers they can respond to, and many other functions. Many genes are active in all cells to direct basic cell functions such as energy production and repair. These are housekeeping genes. During development, many genes direct the formation protein that switch off other genes. These are gene regulatory proteins essential as developmental processes. The majority of genes with a cell are switched off within, so that only a very limited amount of the total genome in each cell is operating to produce the specific function required. The orderly switching on and off of genes during development ensures that cell division, death, migration, and differentiation take place in the correct sequence at the correct time and in correct place to produce the complete functioning organism that is human. Another important concept to understand is that when cells embark down a pathway of differentiation, there is usually no going back to an earlier, less differentiated stage. Cells therefore become much more restricted in what they can become as development proceeds until eventually they are fully differentiated into a specific cell type. There are cells that retain some of their embryonic characteristics and can differentiate into several cell lines. These cells are useful for generating new cells to replace cells lost by normal wear and trauma disease stem cells. As cells of the embryo differentiate, they initially form specific germ layers from which certain cell types, but not others, can differentiate. Four germ layers develop during pre-embryonic and early embryonic period, each giving rise to a specific set of cell types. Cells originating from two or more different germ layers are usually required to form tissues and organs. The development of the right number of cells of each kind, their aggregation and differentiation must be coordinated precisely, signaling between different germ layers. Signaling between different germ layers is described as interaction. Often one germ layer determines what another germ layer does by causing a change in shape, movement, or function. This process is inducting. The determinant germ layer produces signals. The determinant germ layer produces signal molecules that act on one recipient germ layer. The inducing cells may not show any obvious change as they act on other cells, but may then exhibit marked change in their own behavior. This change is often induced by the cells they originally induced, and when germ layers take it in turns to induce each other reciprocal interaction. Now, the fundamental processes involved in the embryological development and growth have been outlined. We will apply them to examine how development takes place in the first few weeks of life. A whole raft of arcane terminology is used in many textbooks to describe embryos and the nervous system. As you will see, the front and back of head and tail end of an embryo can be distinguished from a very early stage. Everything can therefore be referred to the anatomical position, abbreviation, the need for obscure terminology. And this lecture, reading the book, has ended.